In this session, we are going to discuss an extremely important topic in laboratory operations and scientific research, namely principles of scientific documentation and data recording. No matter how advanced an experiment is, its value is lost if the results are not properly documented. Scientific documentation forms the foundation of research integrity, quality assurance, regulatory compliance, and reproducibility. In simple words, every experiment tells a story, but that story can only be understood if it is recorded accurately and completely. Throughout this session, we will learn why documentation is important, how data should be recorded, the ALCOA principles of data integrity, proper corrective methods, record retention, and the role of documentation in scientific credibility. Let us begin. Scientific records are much more than just writing notes. They serve as tangible evidence of all laboratory activities and experimental work. Whenever an experiment is performed, the laboratory notebook or electronic record becomes proof that the activity actually occurred. Documentation also demonstrates the approved protocols and procedures were followed correctly. For example, if an auditor visits a laboratory and asks how a particular result was obtained, the answer should be available through documentation records. Scientific records also provide legal protection. In pharmaceutical companies, research institutes, and biotechnology organizations, documentation may support patent applications, regulatory submissions, and legal investigations. Therefore, scientific records are not just operational documents, they are scientific, regulatory, and legal assets. One of the most important concepts in scientific documentation is the ALCOA principle. ALCOA is a global accepted framework for maintaining data integrity. Let us understand each component. A for attributable. Every piece of data should clearly identify who generated or recorded it. L for legible. All entries must be readable and permanent. If someone reviews the record years later, they should still be able to understand it. C. Contemporaneous. Data should be recorded immediately when the activity occurs, not hours or days later. O. For original. The first recorded observation is considered the original data and should be preserved. A. For accurate. Data should be correct, complete, and free from errors. Following ALCOA principles ensures reliability, transparency, and compliance with regulatory requirements. These principles are widely used in GLP, GMP, GCP, and other quality systems worldwide. A good laboratory record should answer four important questions. First is what. The record should clearly describe what experiment or activity was performed. Second is when and who. Every entry should include the date, time, and name or signature of the person performed the activity. Third is why. The purpose or objective of the experiment should be clearly documented. Fourth is how. The record should describe the methods, materials, equipment, reagents, and procedures used. When these four elements are properly documented, another scientist can understand easily what was done and reproduce the experiment if required. This is one of the key principles of scientific research. Laboratories use different systems to record data. Traditionally, But laboratory notebooks have been considered the gold standard. These notebooks are page numbered and difficult to alter, making them highly reliable. Today, many laboratories also use electronic systems and digital record keeping platforms. Supplementary records such as instrument printouts, gel photographs, chromatograms, and analytical reports are often maintained electronically. However, these records must always be linked to the primary laboratory record. One important rule is to remember is never record observations on loose scraps of paper. Temporary notes can easily be lost, misplaced, or discarded. Data should always be entered directly into an approved record system. Many people assume that documentation should never contain mistakes. In reality, errors happen in every laboratory. The important thing is how those errors are corrected. Laboratory records should never be erased. White correction fluid, erasers, or scribbling over entries should never be used. These practices destroy the audit trail and create concerns regarding data integrity. Instead, when an error occurs, draw a single line through the incorrect entry. The original information should remain visible. Next, write the correct information nearby. Then, add a brief explanation, your initials or signature, and the date of correction. This process ensures transparency and allows auditors and reviewers to understand exactly what happened. Remember, hiding mistakes damage credibility, but properly correcting mistakes demonstrates professionalism and integrity. Scientific documentation extends beyond writing notes. Many experiments generate supporting evidence such as photographs, microscopy images, chromatograms, spectra, printouts, and electronic files. These records are equally important and must be managed carefully. All supporting documents should be properly labeled and linked to the corresponding experiment. Researchers should also record metadata. Metadata includes information such as sample IDs, instrument details, analysis methods, software versions, and statistical calculations. Without metadata, valuable scientific information may become difficult to interpret in the future. Proper archiving and backup systems are also essential to prevent data loss. Documentation compliance follows a continuous life cycle. The first stage is design. Documents should be designed in a controlled and traceable format. The second stage is distribution. Only approved versions of documents should be available for use. The third stage is review. Regular audits and reviews help identify missing information and documentation gaps. The final stage is retention. Records must be securely stored and retained for the required period according to organizational and regulatory requirements. Compliance is not a one-time activity. It continues throughout the entire life cycle of a study or project. Let us now understand why proper documentation is so important. First, it protects participant safety, especially in clinical and biomedical research. Second, it supports regulatory accountability. Audits and inspectors rely heavily on documentation to evaluate complaints. Third, it enables reproducibility. Scientific findings gain credibility only when other researchers can independently repeat and verify them. Fourth, documentation enhances institutional credibility. Strong records support patents, publications, regulatory submissions, and scientific reputations. In many cases, the quality of documentation reflects the quality of the laboratory itself. Good documentation builds trust among regulators, clients, researchers, and the public. To conclude this session, scientific documentation is a critical component of laboratory operations and research activities. We discuss the importance of scientific records, the ALCOA principles, characteristics of high quality entries, data acquisition systems, proper correction methods, record achieving, and compliance requirements. Remember one very important statement: if it is not documented, it did not happen. Documentation is not merely paperwork, it is the permanent evidence of scientific work, the foundation of data integrity, and the key to regulatory compliance. By maintaining accurate, complete, and honest records, we ensure that scientific discoveries remain trustworthy, reproducible, and valuable for future generations. Thank you everyone for your attention. If you have any questions, kindly post them in the comment section.